So all the slides are being brought up here. I'm Michael Keiser. We have a 5-person team from UCSF at the Institute for Neurodegenerative Diseases, where all of us are actually, though, in different departments, but we're all in the same institute. And so 3 of us are here today, and we're going to try to have all 3 of us present in this 15-minute slot. So what we're interested in is Alzheimer's disease. And as we've already heard about this morning, there is a substantial body of work now bringing to light this idea of prion-like propagation and also the misfolding of certain proteins in this and other neurodegenerative disorders. We're focusing primarily on tau and A-beta. And really, an interesting aspect and also a big, I think, frustration has been that although there is genetic support and evidence for things like A-beta, really we don't have a good sense of the general cellular mechanisms of what's going on controlling this sort of etiology. And so just in the simple schematic as you have here, you can see that there's a lot of question marks on this slide. If you think about the cellular process, not just of the misfolding of the protein, but its propagation from one cell to the other one. Once it's there, how it might be broken down, cleared, what is going on. So the way we're approaching this is really in an interdisciplinary manner. I'm coming from a systems pharmacology approach. This is a new field. It really only was sort of given a name in 2011. And it means a lot of different things to different people. But for me, it's looking at a computational basis to look at the way that small molecule structures take us to drugs and the way that small molecules work at different scales, all the way from the single interaction with a protein through cellular networks. Martin will be talking about functional genomics. And then Dave will come in with behavioral phenotyping in whole organisms and vertebrates, in this case zebrafish. And so one way you could imagine starting to approach this, if you wanted to come at it in as unbiased way as possible, is that you could look at lots of small molecules. And so this is work that Stanley Prusner's group has done, screening over 300,000 small molecules in a cellular assay. Here, the cellular assay, the focus is, on, as I said, on the propagation of these aggregates from one cell to the other. And so out of those 300,000 molecules, which you can select from as broad a library as you can, you look for the compounds that are hits, or at least that are active in some way in this system. And by the way, one point before I go any further I'd like to emphasize is that all 3 of these different approaches we're using here in this system with this question, but they're general approaches. And so the actual work that originally led, for instance, to the similarity ensemble approach, or SEA, was really just looking at unexpected drug activities. But what we can do is we can look at the structure of a small molecule. And so out of that 300,000, say we have one here that was active in the screen. And this is not an active molecule. I'm only showing it just as an example. If you were to look then at all the proteins that it might bind, we know about more than 2,000 of those that are therapeutically precedented. There are known ligands. Now, not all of those proteins have crystal structures or known structures that we could actually try to model. So instead, what we can do, if we want to cast a really broad net here, is look at patterns in the small molecule structures for our molecule versus the molecules that are known to that protein. I'm showing 2 molecules as an example for thymidylate synthetase, or TS. In practice, we might have 200 or 2,000 pulled from the literature. And as I said, there's about 2,000 or more such targets. We're going to run our comparisons computationally across them all, ask where each of these comparisons of a single molecule against a target falls on a random background distribution of what random chemical similarity would look like if you just chose a molecule, and find those that are the most significant. And so from an approach like this, we can create a profile of proteins that any given small molecule is predicted to bind. This profile doesn't have to be perfect, though, because what you can do is, as I said, if we have 300,000 small molecules, there's more than one that's a hit. And from all of the molecules that are hits, we can ask not just which proteins are bound by the molecules that were active, but which proteins were bound by all of those other molecules that failed. And this is an important point, because something that I've seen to be very lacking, or at least I haven't seen much done in so far, is looking in these phenotypic screens and using the negative data, not just looking at the things that worked, but everything that failed too, because that's what gives us discriminatory power to ask which proteins actually drive the outcome. And so there's some statistics behind that I'd be happy to talk about later if you're curious or in questions. And so when we have that in hand, what we can do is build lists of prioritized cellular machinery targets that are underlying what we're seeing in the screen. And so instead of looking at any given chemical structure one by one or having to rely on human intuitions or approaches, we can look at these whole lists. As we do this, we can ramp up the potency of the effect and then recalculate this whole procedure at each time. And just to give you a sense, for this, there's about 4 trillion comparisons that are run. And from this, we see these proteins coming out that are involved in other work, in neuroprotection, tau, polymerization, phosphorylation, cleavage. These are the sorts of proteins that then, again, come from an approach that knew nothing about the biology, but really are driving us to biology that we do know in new directions, too. And then finally, before I turn it over to Martin, who'll talk about functional genomics, the thing that really gets me excited is not just doing this in a one-off way, in a one protein, one molecule, one disease, but asking, are there pairs of proteins that only in tandem work? How many diseases and how many cases of Alzheimer's molecules that are effective would only be effective if they hit more than one protein at a time, and maybe even in the screens right now that are already run phenotypically, that's how they're working. But that's a huge space. And so the calculations that we're doing actually allow us to look at this too, and to enrich, particularly for certain pairs of targets that work in tandem. And so in this example, I'm showing not just tau, but purposely going over to completely different neurodegenerative disorder, Creutzfeldt-Jakob, looking at prion protein scraping and asking what signatures are in common for them both. And that's what this wheel is showing, that some of these proteins paired with other ones actually are underlying the molecules that are effective in one or both of these disorders and screens for these disorders. But really, this brings us to testing the predictions. We test these pharmacologically by known drugs that work this way, but also the next question, of course, is genetically and genomically as well.
Thank you, Mike. And, is my mic on?
Yep.
Okay. So as Mike nicely introduced, in our group, we are really interested in trying to combine different big data approaches to get at the question of what is driving some of the cellular processes important in Alzheimer's disease, specifically tau aggregate propagation and formation, both from biology perspective and also to identify potential therapeutic targets. And one of the ideas that Mike introduced is that it's maybe not one factor at a time that we can find, but really a lot of power from looking at combinations of factors. And in some sense, these interactions between several factors might also explain why the vast majority of Alzheimer's cases are considered sporadic, because maybe it's a combination of several factors that account for this hidden heritability and genetic interactions between them. So Mike's approach is pharmacological. I'm taking a genetic approach, and basically I don't have to introduce CRISPR-Cas9, but it's been really a very powerful tool to manipulate the human genome and human genome function. The naturally occurring Cas9 is a nuclease that, in a programmable way, binding to a small guide RNA, introduces cuts in the DNA. And generally, they are repaired by non-homologous end joining in cells. And so this can actually be used as a perturbation tool for pooled genetic screens, where you rely on error-prone repair to disrupt genes and specifically indels that introduce frame shifts. And several labs have used this as a screening tool. So when I was a postdoc in Jonathan Weissman's lab at UCSF, I helped to develop a system that is using a variation of CRISPR-Cas9 to do both loss and gain of function screens, and we basically termed these CRISPR-I and CRISPR-A. We use a catalytically dead version of Cas9 that no longer cuts DNA, but that can still be programmed to bind specific DNA loci, and that way recruit functional domains such as transcriptional repressor domains or recruit transcriptional activators to either repress the transcription of endogenous genes or activate it. And so I don't have the time to go into all the technical comparisons, and this is actually published work, but it turned out to be a very powerful system to really identify hit genes. It's vastly superior to RNAi-based approaches that I had also worked on, and definitely highly comparable to CRISPR cutting. But the nice thing is CRISPR-A allows us to do gain of functions. And for example, in a therapeutic context, ask, are there certain factors that would have a beneficial effect in cell-based models? to get at the interaction questions, one of the things that people have done in classical genetics for a long time is to look at gene combinations to identify pathway interactions between genes. So if you have a quantitative phenotype like fitness, for example, you can ask what are the quantitative phenotypes of mutants A and B. And then you can ask, what do you predict for the double mutant AB? There's a certain model that you expect for most genes that don't interact in a specific way. From population genetics, that's just a product of the two individual phenotypes for fitness. But things get more interesting if the expected double mutant phenotype is different. So you could have less than severe interaction, for example, if A and B act in the same pathway. So inactivating B inactivates that pathway already, so A on top of that has no strong additional effect, so that will be classical epistasis. Vice versa, you could get a very strong synergistic effect of perturbing two genes, for example, if A and B act in parallel pathways and can compensate for each other's loss, and synthetic lethality would be the most extreme case of that. So we can now do this, and this has been done by the Weizmann lab and others in yeast. And we can do this now in human cells as well to really perturb very large numbers of gene pairs and use the quantitative double mutant information to gain pathway information. Again, it's published work, and I don't have the time to go into much detail. How do we apply it in this project? So the first thing we did when we were funded by the Allen Foundation was to establish a cell-based reporter for tau aggregation and tau propagation that we could use in genome-wide screens like this. And the first work was all done in HEC293 cells, although as I won't have time to show you today, we are now building this in iPSC-derived neurons, and we have the whole CRISPR-I system running there as well, and just conducting a first pilot screen. But the first work we did in a HEC293 cells, where we following a similar model published by Mark Diamond, expressed two versions of tau fused to two different fluorescent proteins that can act as a FRET pair. Normally, there's no tau aggregation, there's no FRET, but you can expose these cells to recombinant tau fibers that we make from E. coli and fibrilize in vitro. And if you add them to the culture medium by these unknown uptake pathways, they get into cells and trigger aggregation of the reporter that we can measure. We can see it by microscopy, and we also get a very strong FRET signal. And that's important because that allows us to use flow cytometry to separate out cells that develop aggregates from not. And we can titrate in the amount of fibrils so that we get, say, 50% of the cells in the population developing aggregates. We can do this on the background now of our reporter cell line, into which we introduce genome-wide guide RNA libraries, and then we can separate out by FACS cells that developed aggregates or not, and ask what are the genes or gene combinations that really increase or decrease your likelihood to develop these aggregates. And we completed the genome-wide screen in 293 cells and found several genes of interest, some categories overlapping with the genes that Mike identified with his approach, so that's obviously very exciting, and we're prioritizing those. Some in expected pathways having to do with protein folding, degradation, trafficking, acting actually in both directions on this process. And so the next steps that we're in the process of doing are biochemically validating how they work by measuring aggregation biochemically and characterizing the types of aggregates, subcellular localization that we get. We are validating them in iPSC-derived neurons, looking at endogenous tau, and we are next hoping to do a CRISPR-A screen for gain of function, as well as a genetic interaction map to understand connections between these. And so with that, I'll pass it on to Dave.
So we use zebrafish because they're vertebrate animals with complex brains and behaviors, but they're small enough to fit in 96-well plates, and they live in water, so it's easy to expose them to drugs. And that's important because most neuroactive drugs were discovered based on unexpected behavioral phenotypes. Alcohol, cocaine, pot, and opiates, all discovered based on unexpected phenotypes, and not just natural products, but the prototypes of most modern psychiatric medicines, the first antipsychotics, antidepressants, anxiolytics, all discovered based on unexpected behavioral phenotypes. So behavior-based drug discovery is effective, but it's also relatively low throughput. In vitro assays are higher throughput, but they sacrifice the physiological complexity of intact living animals. So a major premise of my work is that zebrafish can combine some of the advantages of high throughput screening and complex behavioral phenotyping. So this is a movie of a 96-well plate filled with zebrafish. There's 8 animals per well. And when I play the movie, you can see how these animals will respond to these purple light stimuli. You can follow the bouncing red ball to indicate the timing of the stimulus. The stimulus turns on, and you get this robust behavioral effect. When the stimulus turns off, the animals start to calm down and relax. You can imagine what might happen when that stimulus turns on again. The animals start to move again. Stimulus turns off, the animals will stop. We look for these robust and reproducible patterns of behavior that we can assay in 96-well format in a way that scales really nicely. We can routinely, with one machine, look at 2,000 compounds a day or 20,000 fish per day, any gene or compound that we're interested in. So we've developed some software that will quantify the patterns of behavior that we see in each well. And we don't just look at one behavioral assay, but a whole battery of behavioral assays to get a high-resolution profile of how zebrafish respond to different stimuli, different compounds, and different genes. Given a drug-treated behavioral profile, we can use that as an input query to blast our database of 10 behavioral assays and 30,000 compound treatments to try to identify novel compounds that cause phenotypes of interest. In this project, we're applying this approach to Alzheimer's disease by making transgenic models of zebrafish that overexpress human tau and then looking at a variety of biochemical, cellular, and behavioral phenotypes. For example, this is, the zebrafish is transparent at 5 days old. If we just look in the trunk, you can see that these animals, you see some nice neurites and axons expressing GFP tag tau. In this bottom panel, you can see, again, some axons, but also, these neurons are expressing this mutant tau tagged to GFP, and I think you can probably see some spots. We think that these dots represent aggregates and the early stages of neurodegeneration. Ultimately, we're hoping to identify behavioral phenotypes in the disease models and then screen for compounds that normalize these phenotypes to potentially treat disease. So together with Michael and Martin, we're working to integrate technologies in systems pharmacology, functional genomics, and large-scale zebrafish phenotyping to understand some mechanisms of neurodegeneration and discover compounds. Thanks.
So I think the combinatorial approach is absolutely great, but I wonder how you deal with the challenge of the fact that you're dealing with larval zebrafish, which is a developing animal, to model a neurodegenerative aging-dependent disease, and whether the fact that you overexpress a mutant tau developmentally is going then to bias the kinds of things you pull out in terms of drugs and genes that are going then to take you to a set of fantastic answers to a completely different question, which is neural development as opposed to it.
Yeah, I think that's totally possible. There's lots of potential differences. There's lots of differences between humans and zebrafish, and there's lots of reason why some of the hits may be, say, zebrafish specific and not translate to human disease. On the other hand,
no, I was referring to the fact that the fish you use is a developing animal, and it's not an aging animal.
Right. So to the extent that we can recreate AD-related phenotypes in the fish, I think we'll take those phenotypes as far as we can. And you're absolutely right. We would want to follow up with any hits we find in other systems, in human cells, in older animals. And I've talked about larval fish, but of course you can also follow animals as they age and test these in older animals or mice. But it's a discovery engine, and yeah, we assume that not all hits will translate.
But just to add, in addition to that, is with the 3 different approaches, each one has its own limitation or bias, and they are different limitations and biases. So in the example in a behavioral assay in high throughput, we have the question of development. In the example of the cells, where you're looking at the propagation, as I mentioned earlier, in the human cells, there's a completely different bias that we're looking at there. It's precisely because of these different approaches that we are triangulating, because we are not attempting that any one system will fully model it.
And I have a second question. I mean, this is an unfair question to you guys particularly, but generally all the Alzheimer's talks so far, one fascinating thing about them is that none of them address this sort of standard hypotheses, which is great. So they're trying to find new arguments, but none seem to deal with the, and this is a question to everybody who, to deal with the basic fundamental physiological function of APP and tau. So can we disregard what these genes normally do in our search for how mutations in them or otherwise modifications of them are relevant to the disease? Would perhaps an approach like yours actually address that in some way?
I mean, it's a great question, and I think it's a really fundamental question that applies to all neurodegenerative diseases that have protein aggregates, right? Is it a loss of normal function, a gain of toxic function, or a combination of both, right? For example, in alpha-synuclein, which aggregates in Parkinson's disease, I think the knockout mice are fine, right? So it seems to be actually somewhat unrelated to the natural function of the protein, and clearly we are all studying tau aggregation in a system where we don't look at the normal function either. And you're absolutely right, we might be missing an aspect of it. But having said that, if you think that aggregation is something also that prevents it from doing its normal function, then studying what underlies and controls aggregation is going to still be meaningful, right?